Strain. I am recording in progress. I've now got to make sure that it's almost like that moment in Big Brother. You know, you are being recorded. Please be careful what you say. My name is David Strain. I'm a clinical senior lecturer working in the Diabetes and Vascular Research Centre. And for the last couple of years, in case anyone's missed it, there's been a bit of a virus going around. And I've been doing a fair amount of clinical work with that. And what I'm hoping to, or what I'm going to be talking about for probably the next 25, 30 minutes or so, and then throw it open to questions and answers, is about the sequelae of this, about long COVID. You've heard lots of conversations about the treatment of acute COVID, but I just want to take a quick moment to talk about long COVID. I'm hoping you can all see my slides. Can you? Good. Okay.
David, can I just say something? Sorry to interrupt you. I can only see half your slide. So I've got long and then the capital C, but I can't see any more. Am I the only one that's got only half a slite, or is everybody else?
I can see the whole slide.
I can see the whole slide.
I wonder if you've got it oh, okay. I wonder if you've got it expanded. If you just
I'll just click on it.
Yeah.
I wonder whether if you've got people's faces on your screen, you might be able to move. It might be because you've got them on the screen as well. You might need to move them. Is there an arrow you can click on, or on the top right corner, click on view, and you can change?
Don't worry, it's fine. I don't want to hold things up. It hasn't happened before, so I'm not quite sure what to do. But although I do this kind of thing quite a lot. Just carry on, David, that's fine.
Linda, if you right click on the slide, an option should come up that says fill screen.
So if I click on,
if you right click it,
hang on.
It might be an option saying view options at the top.
Yeah, in fact, it's at the top under view options. You see it says you are viewing David Strain's screen, and it says view options.
Oh, I see. Fit to window.
50%,
100%. Fit to window. Let me just try. Sorry if I'm holding things up.
That's okay.
So I need fit to window. Do I need fit to window? Is that what I want?
Yes.
Yes.
No, that doesn't work. Fit to window. I tried 100%. That doesn't change it either.
50% changes it.
Okay, we go 50%. That's just a smaller version of the half page. Please don't worry about it.
Oh, really? You can still only see a half page. That's bizarre.
I know. Now I've got a smaller half a page than I had before. But please
just go back to fit to window then.
Okay. But it's still only half a page. So view options.
Maybe look at your view in the corner and put standard. Right up in the top right hand corner, there's a little box that says view. And what have you got that on?
Show manage participant, show chat.
Put it on standard.
Don't worry. Exit full screen, annotate, hide video panel. Please don't worry about it. Honestly, I'd hate to hold you up. Don't worry. I can make up the rest myself. Don't worry.
It's going to be more talking than it is about the work.
If there's anything I don't understand, I'll just ask you later.
Perfect. Or just stop me there and then if there's things that's missing.
And also, if I can get the slides off you, David, I can always share them with the reps anyway.
Yes, that's fine. Don't worry. I can always click on the recorded version, so please don't worry about it.
So, so basically what we're going to be talking about is long COVID now. For those of you who are uncertain about what long COVID is, you are probably in the majority, because even amongst people working in the area, or people who are working in the field, there is discrepancies or disagreements about what constitutes long COVID, what the underlying cause of long COVID. In some cases, that long COVID even exists as an entity in its own right. But actually, the history of post-viral illnesses is not a new thing at all. Actually, the best documented cases of the post-viral illnesses is after the big pandemic in 1918 to 1920 of flu. Many of you will have either seen the film with Robert De Niro or Robin Williams, or even read the book by Oliver Sacks that he wrote in 1973, talking about the discovery of levodopa for a group of patients with a Parkinson's disease-like syndrome. The bit that's actually missed in the film, but the book goes into great details, is this is not Parkinson's disease per se. What Oliver Sacks's discovery was about was encephalitis lethargica, which was a Parkinson's disease-like syndrome that affected people after that original pandemic in 1918. And that led to the discovery of levodopa, which is the core treatment for Parkinson's disease today. I have to say, if you haven't seen this film, you really should. It's just a fabulous film talking about medications discovery and how a lot is done through trial and error by the observations. And this highlights the fact that the experience of new disease is actually probably one of the most exciting things that can happen to any medical scientist during their career. Because effectively, to land right at the ground zero of a brand new disease that we don't know what causes it, we don't know why it exists, we don't know how it is going on, is a massive opportunity. Whilst at the same time, it can be one of the most frustrating things possible for the patients that we serve who've got this disease. Because to go and see their doctor, to go and see their healthcare practitioner and be said, yes, you have a disease, but we don't have a test for it, we don't know what causes it, and we don't know what to do about it, is also one of the most infuriating things, both for the doctor and mostly for the patient that's suffering with it. Now, long COVID as a phenomenon was first described in a publication back in August 2020. Actually, we first started to see that there was something going on around June 2020. This is months before there were any official words on it. And the reason that we started to see it going on is one of my clinical roles is I was providing the support for people who live with myalgic encephalomyelitis and chronic fatigue syndrome. I was the physician who spent once a month, spent an afternoon going through those patients to see if there's anything we could do for it. Now, many of you will have heard of chronic fatigue syndrome, or ME, myalgic encephalomyelitis, and that presents with a very, very similar condition. The reason we started to see this going on is because these patients who'd experienced COVID in March and April were then being referred to the chronic fatigue service. Now, the name long COVID itself has been hugely controversial in its own right. Because although you speak to the media or you speak to many doctors working in it, they'll call it long COVID. Long COVID is actually not the official term for it. The official term is both new or ongoing symptoms post-COVID or post-acute COVID syndrome. And they are defined at 4 weeks, when if you have symptoms beyond 4 weeks, you've got ongoing symptomatic COVID-19. And then you've got post-acute COVID-19 syndrome when the symptoms go beyond 12 weeks. Now, they are the official diagnoses that you'll get at NICE, that you'll get from your GP's database, that the World Health Organization represents. However, the patient preferred term stays as long COVID. Now, this is a patient preferred term that was originally put out in May 2020 by Elisa Peregro, who was an archaeologist. Actually, she's an archaeology student at UCL, but she comes from Lombardy. She caught COVID in the very first wave in January 2020 and described her symptoms as ongoing on Twitter, of all places. Her tweet went viral, went round the world, and this hashtag of long COVID stuck. That's where the name came from. Now, the participants that we've got working with us in many of the studies much prefer that term of long COVID to the official WHO post-acute COVID syndrome. And the reason for that is very, very simple. Post-acute implies, firstly, that something is over, and secondly, that it's in some way related to the original severe disease. What we see in long COVID is actually that firstly, it is not over. Currently in the UK, there's about 450,000 people who are experiencing this more than 2 years after their initial infection. That's more than 2 years after they caught COVID, which means we are just looking at people who caught COVID in that very first wave from March through to July in 2020. The second is the reference to the acute COVID. Many patients had very mild original syndrome. The severity of post-COVID syndrome or long COVID is not directly proportionate to the original infection. Now, don't get me wrong, if you had a very severe original infection, then you are more likely to have long COVID. But only about 5% of the population had a severe original infection, whereas 95% of the population had a relatively mild condition. In fact, about a third of the population probably don't even know they had it in that first wave when the virus itself hadn't gone through the multiple variants and therefore wasn't causing the same problems that it causes now. Therefore, the vast majority of people with long COVID have only had a mild original disease, and their symptoms have started months later. Now, this question is talking about what are the symptoms, and actually the symptoms give way to understanding of how this virus works and actually how this virus came about to cause so many problems with the acute illness. And if you will bear with me, I'm going to show you some pretty technical slides. This is actually going through the basic physiology of how we fight infections. And so at the end of the next 4 slides, your knowledge of how we fight infections and how COVID has influenced that is going to be probably greater than many of the doctors that you'll be seeing. And it all starts about how this virus enters our cells. Now, it enters our cells using something called the ACE2 enzyme, okay? And I have to admit, the vast majority of doctors hadn't even heard of the ACE2 enzyme before COVID pandemic hit. But the ACE2 enzyme is present on every single cell in your body, because the ACE2 enzyme is actually part of your body's defense mechanism. You see, if anything goes wrong with the body, what the body does is, well, it's got this safety system that is supposed to help protect your healthy system. You produce this angiotensinogen, angiotensin 1, and basically it floods your body with toxins, and your body gets filled with all those toxins, and then it gives your healthy cells an antidote to those toxins. So flooding your cells with toxins goes through the ACE pathway. It causes fibrosis, it causes inflammation, it causes oxidative stress, one of those terms. It's just a generic sort of term that says something unhealthy is going on here. And then it gives all of your healthy cells the antidote to this. So it causes anti-inflammatory, antioxidants, it causes blood vessels to open. And actually what should happen in health is that you have this balance between toxins and the antitoxins, and your healthy cells are protected by it. Now, there are a lot of diseases where this balance is out of kilter. Diabetes is one of those diseases where you see much upregulation of the injury side and downregulation of the tissue protection, and the overall impact is the fact that we see of diabetes on the body. What we saw in COVID was the COVID virus was using your protective enzyme to get into your cells. It therefore turned off the protective enzyme. And as a result, what we saw is a reduction of your tissue protection and an upregulation of your tissue injury. Now, in acute COVID, that is why people who had pre-existing vascular disease, such as heart disease or peripheral artery disease or strokes, or also people who had diseases that are associated with vascular disease, such as obesity and diabetes, had much worse outcomes in acute COVID. because their body was already shifted towards the harmful side, and therefore in that acute phase, it was pushed much, much further. And that's why COVID was causing more of a problem for those with vascular disease than it was for pre-existing lung disease. So COPD and asthma don't trigger this pathway. And what we did see quite early on in COVID is, unlike other viral illnesses like flu and pneumonia, the biggest problems from COVID came to those with vascular disease, not respiratory diseases. However, the position that we're left in now is that these enzymes are absolutely everywhere in the body. Every single healthy cell should be expressing this. And therefore, long COVID can be affecting any cell that is expressing this virus. And that's how that we end up with this hodgepodge of a disease. It can affect people pretty much through any organ. People will experience symptoms in the brain, in the heart, from the lungs, from the pancreas. We've seen a 2.5-fold increase in the new cases of diabetes in people who've had COVID. And that's because you get direct inflammation of the pancreas during your acute infection. We've seen people have progressed to chronic kidney disease in a very, very short time. So they've gone from completely healthy kidneys to actually needing some renal support. And what we really don't know is how long these symptoms are going to last, because we don't actually fully understand what the cause of this is. We do see that symptoms seem to cluster. And what you see is there is the core of fatigue. Now, the fatigue that people describe is not just the tired feeling that you get when you've got a new baby in the house and you're not sleeping. The fatigue is termed post-exertional malaise. That is, it is disproportionate to the amount of activity you've been doing. So you may walk 100 yards and you feel as if your body has just run a marathon. Similarly, the fatigue is not necessarily related to the exercise that you've done. So there are some people who will do major exertional brain exercises. They will be dealing in webinars. They may just do a really tough crossword, and then they will feel physically tired afterwards. That seems to be a core element. Actually, in the surveys we've done, it's about 90 to 95% of people will describe some sort of fatigue. And then there are symptoms that sit around that, and they do sit around it in a relatively predictable way. So brain fog, for example, this inability to concentrate, the inability to focus to the end of a sentence, the difficulty just following a conversation, is one of those key symptoms that a lot of people present. And if you've got brain fog, you are more likely to have a fever and or chest pain. If you present with fever and brain fog, you are more likely to end up with a headache. But it is unusual to see people complaining of breathlessness at the same time as brain fog without them relating. Similarly, chest pain, and it's a very similar chest pain to we experience in myocarditis, inflammation of the heart muscle. Chest pain does sit with brain fog. Chest pain also sits with palpitations, but it is very unlikely for somebody to have palpitations and a headache without being connected through those pathways. And so, although it can present in anybody, it does seem to have regular, predictable clusters that it sits to. Now, actually, this is quite an old slide. I wrote this quite early on in the pandemic, and there was a paper published just last week by Tim Spector from the UCL, from the Zoe app. But actually, he's now got the evidence that something that we've been saying for a long time based on the Zoe app, that these clusters do seem to have relatively predictable elements. There is one bit that fits right across the board, and that is an anxiety and low mood. Now, the anxiety and low mood is very, very difficult to try and tease out. And what we don't know yet is whether the anxiety and the mood dysregulation that fits with long COVID is an independent symptom in its own right. And if you think about it, anxiety and mood is just another brain function. If one bit of the brain's not working, then the anxiety and mood centers might be not working. Or whether this is just a response to the fact that somebody who was otherwise very fit and healthy is now all of a sudden in a position that they've got major problems, and they've gone from having a complete health life to being in a chronic disease status without going through those previous steps that many people get to in order to go from being fit to being living with a long-term condition. And there is a considerable amount of work. Actually, it's part of the cohort that we're working with, and Nisrin Alwen is leading on it, to determine whether this anxiety, whether this low mood that people are getting in COVID is a direct response to the long-term condition that they're living with, or whether it's its own right, its biological activity that's going on. So what are we up to in the research for this? Well, what we want to do is find a potential treatment, but we can only really start looking for treatments properly when we start looking at the underlying causes. Now, so far in the UK, we have invested about £50 million of NIHR money, and there's about a similar amount of money that's been coming from other groups into looking at research. Now, that sounds like there is a lot of money, and it does sound like a lot. However, £50 million with 2 million people who've got the disease represents about £25 investment per person. Now, £50 million in research term doesn't go a very long way. Actually, the US have promised $1.4 billion to research this. And although they've promised $1.4 billion, absolutely none of that has started the research. That's nowhere near as impressive as it sounds. And as a result, we are still very much in the embryonic phases of it. So what I'm going to show you is a few pictures that are just demonstrating where the research sits at the moment. I'm going to be explaining what it actually means. But then the last few slides will be highlighting the information that we've got, for while we're planning to do the research. And actually, sorry, the reason I was temporarily distracted a moment ago is some of the data that I've been trying to get for the last week to show for you has literally just come through on my desktop. So I will be flipping presentation in just a moment to the exact numbers that we've got here in Exeter. It's always the way you spend weeks going for it. It comes at just a few seconds too late. So the first thing to say is the immunology. So Danny Altman's group in Imperial College in London have done this. This is a really complicated slide, and to be honest, it took me about an hour and a half to figure out what this was actually saying, because this is the overall picture that Danny Altman believes is self-explanatory. But what is showing is looking at the immunoglobulins. So your immunoglobulins, they're the bits that respond to an infection. They're the bits that we are measuring when we're looking at the antibody response. And the immunoglobulins in people who develop long COVID have a different pattern to those who are not responding to long COVID. Now, at first, there was a question then, is long COVID causing an autoimmune disease? Are we seeing specific antibodies being created to that COVID virus that is then continuing to fight the body afterwards? However, that doesn't appear to be the case. So what we've got here is looking at people on the left who don't have long COVID, and on the right are people who do have long COVID. Up and down in this picture, what you see is the total amount of antibody that they produce. And what we can see is actually, although they were looking for specific antibodies that could cause an immune response, what they demonstrated is people who've got long COVID actually mounted a poorer immune response. And when they tracked it down, there was one specific immunoglobulin that was seen to be deficient. The one specific immunoglobulin is this one here. It's in IgG3. Now, IgG3 is an immunoglobulin that we all have to some degree, and that is in part responsible for protecting our body against long-term infections. Other diseases that have been identified to have low levels of IgG3 are ME, myalgic encephalomyelitis. People who've got long-standing Epstein-Barr virus, glandular fever, have got low levels of IgG3. It's also been recognized that if your level of IgG3 lowers, that is a trigger for you developing shingles. And we know shingles is a virus that hides somewhere in the nervous system. And then if you get compromised for any reason whatsoever, the virus comes back to life. And that's when we see shingles. Shingles will hit us at a time that we are otherwise low. And one of the precursors of that is a lower level of IgG3. What this paper tells us is that long COVID is most likely to represent the virus finding somewhere to hide in the body, and then from time to time coming out and expressing the symptoms. Now, that could explain why people have this in ups and downs. People can have a period where they've had lots of rest, lots of good quality sleep, and as a result, they feel much better. But then something hits them, they overexert themselves, they do something extra, and then for days afterwards, they are hit back. Because
David,
that exertion...
David, can I ask you a question?
Yeah.
But you said that IgG3 was lower in those people who had shingles, but on this diagram, IgG3 is the same in those who have primary, with or without long COVID.
So the IgG3 has been known to be knocked back in this. What we're looking at here, this particular group there, is the subgroup. Oh, I've just hit the wrong slide. Let me just go back quick. Sorry. We've got the amount of IgG3 here is basically the IgG3 is the thing that's being produced in response to the chronic infection. So
those individuals without packs on the left in that square that you just pointed at.
Yep.
The red's lower. So without packs means without long COVID, does it?
These people don't have long COVID.
So, and they've got low levels of
they've
got low levels of IgG3, whereas the people with long COVID have high values of IgG3.
So what we've got in this graph here, this is the total level of IgG3.
Mm-hmm.
Total level of IgG3 is lower in people with long COVID.
No, it's not. It's the same.
Total levels of IgG3 is statistically significantly lower in people with ME and chronic Epstein-Barr virus, and also with shingles.
Okay.
Yep.
Yeah. Okay.
Here we have a trend that it's lower.
Yeah.
Probably the wrong picture to be showing, but the trend that's lower. Danny's then gone and done a subgroup down, and what he's done in the subgroup transmission is taken out people who've got long COVID, which is just the residual infection, the residual symptoms of when they had severe COVID. So what he then did was take out them there. He then looked at a breakdown of what this IgG3 was acting against, and the IgG3 in people with long COVID is acting against spike protein.
Yeah, okay. So that big, you can't see my, so in that square, there's one without COVID, long COVID, which is really high in the purple there. Whereas in those whereas in the square that haven't got COVID, that have got long COVID, then the purple is much lower.
Yes.
So is the summary, one of the elements of IgG3 is lower, but not all of it.
One of the elements of IgG3 is lower. And this does appear to be
The bit
the
that
same
attaches the
as
spike protein.
yes.
Yeah, okay. Thank you.
Yeah. So the overall suggestion, and I'm stressing that this is a suggestion, not a certainty by any stretch of imagination, is that people who get COVID have a pre-existing lower level, or a subgroup of people who get COVID have a pre-existing lower level of IgG3. As a result, they don't clear the COVID properly, and therefore that opens up the COVID or the coronavirus to hang around somewhere in the body. And then from time to time, the virus is reactivated, and all of the symptoms that you may expect to get in that acute infection would be recurring. And because everybody experienced that infection slightly differently, it will present differently within individuals, but it will present consistently within an individual. So for you personally, the weakest bit of your body is the fact that you are you've got a bit of inflammation in the pancreas. Then actually every time the virus is resurgent, then what we would expect to see is that's the bit of your body that would be affected. And that's a theory, that's one of the hypotheses at the moment. Now, the place that it was suggested that that's most likely to be active is either neural tissue, and the neural tissue, the brain tissue, that was based around a case series of people who had brain biopsies of who had died of other things, but had had long COVID. And in those brain biopsies, they found evidence of the virus in people who had long COVID still in that brain biopsy. However, that was a case series of 13 patients. And in a match cohort of people who'd also had COVID and didn't get long COVID, 8 of the 12 in that matched population also had residual viral presence. So it is very difficult to say that that virus is causative when, you know, 2/3 of the population who don't have long COVID also have that virus.
Have your lunch if you want.
I can't because it's cooked.
Now, the other place where people have suggested the virus may hide is in the gut. And the gut microbiome has become a hugely interesting area for research in up and down the country. And actually, we've contributed a fair amount to this. However, this was the first suggestion that that may be the case. What they looked at here is the gut microbiome in people who got long COVID. And what they demonstrated is that in that gut microbiome, there was less diversity, as in the total number of bacteria, those healthy bacteria that Yakult and Actimel tell us about so frequently. The total number of different types of bacteria was reduced, and the total quantity was reduced. The problem is that this was done on an incredibly small sample at baseline, 14 people. And the gap between people at baseline is very, very small. 14 people, it is very, very difficult to draw meaningful conclusions. They then have a much bigger cohort of people at follow-up. And at follow-up, the differences were substantially greater. The differences in the gut microbiome were substantially greater between those with long COVID and those without long COVID. And the authors of this particular paper said that this was very clear evidence that the virus had taken up residence in the gut, and that was affecting the gut microbiome, and that the symptoms were being driven by the fact that this gut microbiome that you are absolutely dependent on for good quality health was deranged, and that gut microbiome was a big driver of the fatigue that people were experiencing. And it sounds like a really plausible theory until you start looking at the one thing that regulates gut microbiome. The most important predictor of your gut microbiome is what you put in your mouth. If you put healthy food in your mouth, you will have a more healthy gut microbiome. If you put unhealthy food in your mouth, you'll have a less healthy gut microbiome. Won't change completely, but it will have a big impact on what's going on. It's been very clearly demonstrated that gut microbiome can change relatively quickly. Actually, we are using this in clinically for treating Clostridium difficile, that we can change the gut microbiome very, very quickly with a course of antibiotics followed by replacing them with capsules. However, what the authors of this paper failed to comment on is what happens to the diet of somebody who's got long COVID. And this is actually not been very well studied. People haven't actually asked what them, but what it would be relatively easy to imagine is if you suddenly feel fatigued, you will reach for the high energy, high carbohydrate, high caffeine based drinks. Your overall food intake will change. If you need to get through a session, you will have lots and lots of caffeine in order to do that. And there was absolutely no mention of what happened to their people's diet when they went into this state of long COVID. So although they have very clearly demonstrated that the gut microbiome 6 months after you contract long COVID is affected, what they cannot tell us from this paper is whether the gut microbiome is affected by the virus, which is a driver for symptoms, or the gut microbiome is an innocent bystander for people who've suddenly got a new debilitating fatigue and therefore has a significant change to their input, whether that be their diet, whether it be the nutrition, or just their overall lifestyle. And so this does support that we might need to look at this. However, it's not in any way conclusive. So other anecdotal data is actually some of the stuff that we produced. And I'm going to show you the graph that we've got from medRxiv. This has actually now been published. And this was a questionnaire survey type approach that we did that was just looking to find out what impact did the vaccination have on people living with long COVID. Now, we put this together with 4 of the country's biggest long COVID support groups, and also I reached out to one of the groups in the States and a separate group in Israel. Actually, it was one of my friends from BMA was associated with the long COVID support group in Israel, and there was a big fear that if this was an autoimmune disease, that if you gave a vaccine which promotes an immune response, that you might make long COVID worse. And that's not an unrealistic assumption. We have seen that other vaccinations have made some diseases worse if the disease was an autoimmune basis. The best example of that is psoriasis. We know that psoriasis will get worse after some types of vaccination. And lots of people with long COVID were very scared about getting a vaccine, that it would make their immune response worse in order to the vaccine. What you've got here are each of the key symptoms that patients expressed. And what you've got is whether they had mild symptoms or no symptoms in blue, mild symptoms in green, moderate symptoms in yellow, and then severe symptoms were in red. And in the bars, you've got before and after vaccination in each case. So this first graph is fatigue, and you can see that most people have got some degree of fatigue. There was only a very small number that didn't. And then in the post-vaccination phase, that in every single case, they have got fewer symptoms. Now, this was always looking at people who had had COVID for at least 3 months before their vaccination. We wanted to take away the group who were getting better naturally anyway. We know that half of people who've got symptoms at 4 weeks will get better on its own in that first 3 months. But once you've got symptoms for 3 months, then less than 20% of people make a spontaneous recovery. And actually, if you look at the case finding study where you follow an individual through, patients will get waxing and waning symptoms, but the overall trajectory is if you still have symptoms at 3 months, you still have symptoms at 12 months. And that's been confirmed by the Office of National Statistics here in the UK, the Israeli health authorities, and what work has been done in Canada has also shown that if you get to 3 months, you probably are going to have symptoms for at least the next 12 months. So we have, to some degree, minimized the time impact on this. We can't say it's done completely. But what we demonstrated is, contrary to what we were expecting, that symptoms would get worse, 67% of people reported that all of their symptoms got better a bit or a lot. About 25% of the population said the vaccine had no impact on them whatsoever, and 8% of the population said the vaccine made them worse. So actually, we now have divergent groups. We have exactly the same stimulus for people, but we have a different response. And the majority of people said their vaccine made them better. We then wanted to look to see if there was any sort of dose response curve to this. Because actually, if you're going to have a vaccine, we know that there are 3 different vaccines around, and we know that those 3 different vaccines gave different responses. We've got the AstraZeneca vaccine, the one that that most of us will have had for our first jab. That's the one that was rolled out here in the UK. We have the Pfizer vaccine, and we have the Moderna vaccine. That was the first one to be rolled out in the United States. And what we saw is with these 3 vaccines, you get a different antibody response. So the AstraZeneca vaccine gave you a very good response, but actually the Pfizer vaccine gave you about 25 to 30% more antibodies. The Moderna vaccine gave you about 50% more antibodies. And that's one of the reasons that when it came to giving the boosters were all around the Pfizer and Moderna, because by the time we were ready to give boosters, we knew that you got a better antibody response from the Pfizer and Moderna, and that was one of the reasons. The other reason is to do with the adenoviral vector, which we can talk about at another time, but that also did play a part. When we look at the dose response curve, what we did see is the symptomatic improvement as a percentage was proportionate to the amount of antibodies generated by the vaccine, which we use this as a proxy for which vaccine that we got. So that fatigue, people reported on average about a 13% improvement. With Pfizer, they got about a 17 to 18% improvement. With Moderna, they got a 26% improvement. We saw very similar results when it came to brain fog between AstraZeneca and Oxford, and Moderna and then had better. The autonomic dysfunction, the GI symptoms also demonstrated improved responses. And again, this anecdotally, and I'm stressing, this is supportive evidence. It is not conclusive in any stretch of the imagination, but this is supporting the hypothesis that long COVID is caused by a viral that's persistent that is then occasionally triggering the symptoms. And the fact that it's affecting the GI tract so convincingly does suggest that this may be gastric residual symptoms. So on to what studies that we've got going at the moment. There are 2 genetic studies that are underway. We are looking for genetic causes. The Sanogenetic study is one that we're Sano Gold. I'm the chief investigator for that one. That's genetics of long COVID. And actually, that was a very quick and simple test to look at people, look at the genetics of people with long COVID. We are merging the database for that with the Decode ME study. And the intention there is to look for similarities and discrepancies between people who get long COVID and people who get myalgic encephalomyelitis. The intention there is to find out, is there some genetic predisposition towards it? The genes that we're particularly looking at are those that modulate the immune system, the HLA subtypes. But we're going to be looking at this in a whole sort of genome-wide associations that Exeter has got a tremendous track record in being involved in. I am not a geneticist. I just happen to be the person that was there at the time that got the name associated with this, and therefore get to do the publicity for it. It is going to be a long time before these data are ready. We will come back and have a chat about it afterwards. The study that I'm talking most at the moment about is the Stimulate ICP trial. Stimulate stands for the symptoms, trajectory, and intervention for modulation of long COVID using an integrated care pathway. It's a study that myself and actually my good friend from when we were training historically, a very, very long time ago, that is now a professor of cardiology based at UCL in London, we put together pretty much overnight. The call came out saying that they wanted a call to look for platform studies for an interventional study on, the call was released on Maunday, Thursday, with the intention of having the calls close by the week after Easter. So we had 8 days to put this together. Now, fortunately, Amy and I had some conversations about this in the past, and we'd already been talking about, you know, if we wanted to do a study for long COVID, what we should do. We both have an interest in cardiovascular epidemiology. He's actually a cardiologist. I'm working more on the diabetes vascular side of things, but we both have very similar interests, and we were both seeing a very similar picture, and we were talking about what we would like to be doing had it come across. Through my BMA networks, and through Amy's other networks at UCL, we already had most of the connections in long COVID, so that we could pick up a phone over the Easter weekend and produce the relevant expertise. And quite literally in 8 days, we put together a grant application for £6.8 million in order to look at the symptoms, trajectories, and to look at the social inequalities around long COVID. What we have done is put together 3 work packages. The first was going to be a work package looking at the social determinants of how long COVID is currently treated. The second is a study that I'm going to talk more about in just a moment. And then the third is looking at how we may be able to take the learnings of our new disease and our new pathway into other disease areas. Needless to say, I am not having much to do with the service evaluation of what's going on at the moment. That is all being led by Mark Gabay, who is a GP based in Liverpool. Again, he's part of the BMA committee that I chair, and he's a GP who's got a massive interest in social inequalities and system evaluations. So he is leading on that package. Work package 3, looking at the inequalities. Again, Nisreen Alwan, based in Southampton, and many of you will have seen her. She does lots of media work, both TV and Radio 4. She has a regular slot on Radio 4 talking about social inequalities, and she's leading on that package. When it comes to work package 2, I am the clinical lead on that, and this is looking at the interventions.
Hi, David.
Yes.
Can you just tell us briefly about the interventions? Because I want to leave some time for questions.
Yeah. So the interventions that we're looking at are basically, we're doing a whole load of assessments to find out, does that help with doctors? But the treatments that we want to look at are looking at an anti-inflammatory, an anti-coagulant, and an antioxidant, basically. Those 3 pathways that I started right at the outset, that's saying long COVID is triggering the antioxidants, the coagulation pathways, and the inflammation pathways. We are looking at an anticoagulant versus an antioxidant versus an anti-inflammatory drug. The 3 that we're going for is the loratadine famotidine combination that's been demonstrated to have an antioxidative property. Colchicine, currently a treatment for gout, but it is a very, very potent anti-inflammatory and is widely used in ME for the cardiac inflammation, the brain inflammation that's being looked at there. And although we said aspirin when we originally put it in, we've now changed that to rivaroxaban. It's a very common anti-inflammatory, anticoagulant that is already on the shelf. The reason we're looking at these 3 drugs is that these 3 drugs are already on the shelf. They are cheap, they are widely available, and if we can demonstrate benefit, it would be very, very quick to implement those changes. We have got a few other studies in the pipeline. We are looking at working with one of the manufacturers of the antiviral drug, remdesivir, that we use. We use remdesivir in hospital to clear persistent virus. The idea is that we use a 10-day course of it, and they have just given us a verbal approval only yesterday that they are going to be funding this study. So we're just about to start moving into contracting periods that we're going to be giving the antiviral drug to see if we can actually fix long COVID. And then the third group is the Moderna, the manufacturer of the virus, the vaccine. They actually were really interested in doing a vaccine-based study when we first presented it. They then cooled off purely because they couldn't keep up with the vaccine supply in order to get the rest of the planet vaccinated. And the saying for vaccination is all about until we're all vaccinated, none of us are protected. We couldn't keep up with the global supply. They're now in a position where they can keep up with a global supply and supply surplus. So we're actually about to start having chats with them again to see if we can vaccinate people and give their immune response the boost that it needs to tackle them and make this better. And on that note, I am going to stop talking because having said I was only going to talk for 30 minutes, I've just spoken for 45. So I do apologize for that. I'll stop screen share and we'll invite any questions.
Thank you, David. Thank you.
Yes,
Marina.
hello, thank you very much, Doctor. Can I say that in late November of 2019, I fell ill. I am sure, but nowhere proving it, that I had COVID and that it was around long before the Wuhan people owned up. I was awfully ill and it was taken seriously at my GP surgery. I had all sorts of tests and examinations, and all they could come up with the end, it was some sort of pneumonia, some sort of viral infection, etc., etc. By the time I started to get better, it was being acknowledged. It was being acknowledged at that time. And by the time there was the first lockdown, which was much more effective than subsequent ones, I was fit enough to go out for a walk for 2 hours every morning, around about 5 o'clock in the morning, fine. I then got involved in all sorts of studies and volunteering things, including one for this long COVID brain training study, of which I'm a co-sponsor. Do you know about it? I'll ask you again in a minute. It has got full approval, and it was supposed to have started last month, but it hasn't for some reason.
So three different points there. The first is the very, very clever viral geneticists have tracked back that based on the number of variants of COVID there was when it was officially declared in December, that COVID made the leap from animal to human sometime around September 2019. They suggested that the leap, they were really precise. They said somewhere between the 6th and 8th of September. I think that's just showing off, or they're making stuff up because no one can prove it. So it is not impossible that you had COVID then. And actually the testing that when we were doing in March 2020, even the testing that we had in place then wasn't 100% precise for the exact genome. However, one thing that not many people have spoken about is the influenza that we had in the winter of 2019 was a H3N2 influenza. They are a particularly nasty subgroup of influenza that isn't picked up by the conventional influenza A/B testing. That was going around, and actually that's the one that's in Australia at the moment causing a lot of problems there. I don't know if you're aware from the media, but actually influenza in Australia at the moment is caused 184 deaths last month.
Yeah, it was a
staggering amount.
Right. Thank you, Narina. Let me just
It is a quickie, Angela, but it is an important one. because although I got better and I got involved in all these studies and volunteers as a guinea pig for drug trials and so on, in late March, I did actually contract COVID. And it wasn't severe, but by that time, I had 4 immunizations, so the effects were not chronic. But 3 weeks later, I collapsed, and I was very, very ill indeed. And that continued until about 3 weeks ago, when I woke up one morning and I did feel a lot better, but I'm still coughing. I still can't walk from here to there without being breathless and tired out. But as you say, there is so much we don't know. But I am a little bit disappointed that despite volunteering, I haven't been taken into one of your studies as a volunteer.
So the brain training study is not one of mine, I'm afraid. The Stimulate ICP, we literally only opened, we are the first centre in the country to open, and we only opened that last week.
That's
One of the
patient
big,
being booked in today.
yeah. One of the big problems with the clinical trial process is the duration of time it takes. And although for acute COVID, all of the barriers were lifted and everything was fast-tracked, for long COVID, none of that protection has been given. So it has taken us from confirmation of funding to get to first patient, first visit, actually 15 months.
Yeah,
which is
a long time, isn't it? It's a huge amount of time. Let me open the floor to other members of the panel. Any other questions? Alexis.
I'll go for one. I mean, absolutely fascinating and far too much to take in all at one go, and never will understand it properly. But there are so many things to sort of tease out, really. One is your use of language. I love the description of the gut microbiome being deranged, and I thought that was a great description. On that note, in your last slide about the care pathways, it seemed to me that, am I wrong, has that issue of the gut microbiome been lost? Because you touched for a while on dietary and, you know, the healthy food bit that we talked about. Has that gone from your deeper delve into people and their way of life and the social inequalities as well, which is
so the social inequalities and the environmental impact is a massive component, and actually we now have the risk factors for developing long COVID. And if you are from socioeconomic status 5, you are 24 times more likely to develop long COVID than if you're in class 1. So there is a massive divide, and we are quite there's a group of us who do believe the microbiome is playing a role in that. And I was very skeptical of that paper. I have to admit, personally, I do think the gut microbiome is playing a role, but I don't have good enough data that I can put my hand on heart and say that it's there. It hasn't been lost, and there's a company that makes basically a microbiome replacement capsule, and that is based on gut microbiome that is coming from different parts of the gut from somebody else, that's then all merged together, and you take 3 capsules that dissolve at different parts in the thing. There is a company that make that for Clostridium. Do not chew those tablets. Effectively, it is macerated feces that you're eating. Definitely don't chew those tablets. But they make that already for Clostridium difficile, and we are talking to them to find out, would they be interested in at least trialing it in a small group of patients to find out, does it see an improvement in symptoms? It's not lost. It's just these things all take time.
It takes so much time, and one has to kind of, you know, say what your priorities are, right? And it's kind of, you know, it's probably one, you know, the drugs are the easier option at this time, aren't they? I mean, if we could work on the microbiome, then that would be fantastic and not have to introduce drugs. But it's much more difficult. It's a much more controversial area. Annette, did you want to come in?
Yes, so thanks ever so much for that, Dave, and for explaining it in lay terms. A lot of it, you know, so it's sort of more or less understandable. There's a lot there. I'm just wondering, with those graphs that showed that people had improved across all their symptoms after they'd had another vaccination, people with long COVID, they all seem to improve, but they improvements on the graph, I don't know what the y-axis was, that was it 0 to 800, what was that was? They seem to improve quite modestly, and I'm just wondering how you rule out, because you've talked quite a bit about cause and effect, how you rule out a kind of placebo effect from have attention of the researchers and having another injection, particularly around things like, you know, your ability to smell things and feeling short of breath and feeling very tired. They're quite sort of subjective things. Were there sort of lab markers, biochemical markers to back up those symptomatic improvements?
So simple answer is no. There's lots of bias. That was purely a questionnaire. That was a hypothesis generating proposal. That is not evidence in the purest sense. It's just about generating a hypothesis. And actually the question we were answering was for those organization groups is, should they be recommending people with long COVID to be vaccinated? That's all we can say. Symptoms are very much in the eye of the beholder. So, you know, one person's severe fatigue is another person's Monday. And therefore everything is self-reported. And as yet, there is no reproducible blood marker for it. The nearest Doug Kell in Liverpool was working with, and he's got a measure of microclots. I was actually in a webinar, a chat with him last night. He actually now wants to replicate his data because we think there might be a route for testing this. So he's going to hand the protocols, and we may have a test for it, but it doesn't exist now. All of these things are subjective, and I fully accept the criticism. That is not data, that is not evidence, that is hypothesis generation.
Okay,
I'm saying that it's actually been
criticism.
I'll say that it's actually been it's had the highest citation factor of all the papers I've written in the last 12 months.
And it will encourage everybody to have their next vaccination, right?
Yes.
Any other questions for David?
Thank you.
Can I just ask one quickly? Is that possible?
Of course, yes, of course, Linda.
Can you hear me? Because is it okay?
Yeah.
Number one, thank you so much, David. It's really reassuring that so much research is taking place now to help people with long COVID, and it's really reassuring to know that, and that we've got funding, even though, as you say, it doesn't work out as much per capita. Can I just ask you one short question? Forgive me if I haven't understood this. And I did get your full slides eventually. After the first one, they were full slides, so that was okay.
Oh, that's good. That's good.
It was just something I did with the first slide. Did you say that if you have a pre-existing autoimmune condition, you are more likely to get long COVID?
So there are some pre-existing, I didn't actually say it in this point, but there are some pre-existing immune conditions that do lend you to more long COVID. Rheumatoid arthritis, psoriatic arthritis are the two top of the list. But actually predisposition is, firstly, there is a genetic component of it. So direct blood relatives are more likely to share it. But there are more about autoimmune, histaminic responses. So people who get asthma, hay fever, those sorts of conditions, the atopic, the eczemas are more likely to get it. And they are by far and away the most predictive. Second most predictive is actually myalgic encephalomyelitis and fibromyalgia. And they're a disease that are huge controversy about because, again, like long COVID, we don't have a diagnostic test for it. So there are still some physicians that are uncertain about the exact nature of it. And there's a real hope that those diseases will benefit from the research that's coming into long COVID, because just the acknowledgement that there is so much that medicine doesn't yet understand is moving research forward. And I have to say, the number of people that now want to do research because they realize just how much there is that we don't understand is huge.
That's good.
You did mention specifically glandular fever and shingles. And are those, again, predispositions? If you've had those, are you
the
more
reason I
likely to get
mentioned
long
both
COVID?
of those is they are both diseases that are known that the virus itself hangs around and does problems further down the line. So when it comes to shingles, if you get chickenpox, then at some point later in your life, that virus has hung around in your body for all of that time, and then it can be reactivated.
So you
The
just
vaccine against shingles is actually a vaccine against the chickenpox that you had decades ago that stops it coming out. So too with Epstein-Barr virus, it causes glandular fever. The Epstein-Barr virus hangs around for decades, and in some people is a trigger for other diseases, including multiple sclerosis and some forms of lymphoma. We know that that occurs. So the suggestion that long COVID is just the virus has hung around in the body and keeps coming out to play from time to time has lots of anecdotal and supportive data, even though we haven't yet identified it itself.
That's really helpful. Thank you. That made it much
Alexa, yeah.
Just that I don't want to go down a rabbit hole because I don't pretend to understand this. But when you talked about IgG3, I think I understood that that is reduced and creates a part of the major vulnerability to COVID. If that's correct, is there a way of being able to boost that? To firstly diagnose who's got low IgG3, and then something about boosting that bit of the system as a preventative?
So first thing is that vaccine does boost that. Vaccine will give you, being vaccinated in advance of catching long COVID will boost that IgG3. And that is one of the reasons that we believe that the vaccine has reduced the rates of long COVID. If you're predetermined, oh, it's not me. If you're get phased about it, and we saw long COVID from the original variant was about 15%. With the current variant, it's down to about 4%. And the biggest difference there is the fact that we are all vaccinated. If you look at Delta for people, Delta was during the vaccination time. If you were unvaccinated, 13% of the population got it. If you were fully vaccinated, 8% of the population. And that was the same variant. So we believe that vaccine is boosting the IgG3.
That's such a good message, though, isn't it? That's such a really important message that, you know, now if you get it, you're much less likely to get long COVID.
Yeah, and that's something that we do think that hasn't been capitalized on. We think that we should be, particularly for the vaccine, we should be talking about long COVID more. There is a political reluctance to mention COVID because the rhetoric is all that COVID's got away and it's all about recovery now.
Yeah.
It doesn't quite get to my question, which is,
can you boost it?
The whole population, there will be these people who have this low IgG3.
But if they have a vaccine, they'll get it increased.
Yes, I know, but I'm trying to say, what if we just took the whole world and said, right, you all must have this now, IgG3? Is it a thing you could give?
So there are people with severe ME that do get immunoglobulin infusions, and they do report it's beneficial. Immunoglobulin infusions are hugely expensive, and they haven't gone through robust clinical trials yet.
Okay,
it is something that would be worth looking at the future, but actually from a cost effectiveness point of view, the administration of this is massive. And actually the simplest strategy is just get your vaccines. And if your recommendation is going to be get vaccinated anyway, then there's not much point doing a test if you're going to be saying get the vaccine, which is good for you, whether you've got it or not.
That makes sense. That makes sense. Lovely. I'm going to close the meeting now. David, thank you so much for that really interesting talk. It was really great to learn about it. So thank you very, very much indeed. That was really, really great. And thank you to everybody for joining us today. And we'll see you all soon. Thank you now. Bye.
Bye.
Bye, everyone.
Bye.